Hello, welcome to Homeostasis and Cell Transport lecture video number 2. This is the second half or the second part of our video or lecture, excuse me. Now, so we were talking about passive transport before. Now we need to shift into this thing called active transport. Passive transport, a quick review, was the movement of molecules from an area of, this is passive. Remember, I'm reviewing the old concept. Passive transport is the movement of molecules, it's diffusion basically, movement of molecules from a high concentration to a low concentration. Active transport is the opposite, movement of materials from a low to a high. This is like going uphill or up a hill. Up a hill would be going up the gradient. A gradient would be like a hillside. So your hill has a gradient or an angle at which it goes up and an angle at which it goes down. So if you're going up the gradient, you're going against gravity, and it would take energy to do that, basically going uphill. Another team, they might say it goes against the gradient or up the gradient. Same thing. Diffusion was not like active transport. Diffusion was down the concentration gradient, going downhill. So diffusion was passive transport, no energy. Active transport is using energy to move things uphill or against or up the gradient. So this is going from a low concentration to a high concentration. So here's what this picture is showing you. It's showing you a membrane, it's showing you a protein. This protein is what's called a pump, all right? This protein is moving things across the membrane. See how here we have one H plus over here, and we have a whole bunch over here. We have five of them on this side. So this is low concentration. This is high concentration. It is moving things from an area of low to an area of high concentration. It is going up or against the gradient, and this process requires energy. There's the energy right there showing you. Now, I just showed you that picture that showed you a pump. So pumps, if you see the word pump, pumps move things uphill. They move things where they don't want to naturally go. So active transport has to do with moving things where they don't want to naturally go. Things naturally want to go where there's less of them. Well, that's a natural movement of a material in nature. So pumps, like these ones, cell membrane pumps, like the one I just showed you on the previous slide, they basically bind to certain things, right? They bind to certain things, and then they move them across a membrane. So pumps are proteins embedded within plasma membranes, and then they're moving things uphill or against the gradient, so they're going from a low to a high concentration. So pumps, just like this one back here, this is a pump. Pumps move things uphill. They do things actively, so it is active transport. This is specifically what's called a sodium-potassium pump. This is a pump that we have. We have these in our nerve cells, and these pumps are how our bodies create electric currents and how our nervous system works. So sodium ions, NAs, have a +1 charge. Those are sodium ions. They have a +1 right there. There's the plus. Potassium ions also have a +1. So we need sodium and potassium in our diets so that our nervous system could work correctly. We need them for other things, but specifically for these nervous system cells. Our nervous system needs to create an electrical charge. So since these particles are charged, sodium and potassium, if we move different numbers of them around, we can create a difference in charge, which would create electricity like within our bodies. So how does it work? Sodiums have a +1 charge. All right. So what this picture is showing you, these are 3 sodiums right here. They attach to this pump or this protein. 3 sodiums attached and 3 sodiums are pushed out according to this picture. All right. So +3 just left the cell. All right. Potassium ions, 2 potassiums are moved into the cell. So +2 entered. +3 left, +2 entered. There's a difference in 1. So 3 left and 2 entered, which means there's a difference in +1 overall. So the inside in this case lost +3 and gained +2. So overall it lost an extra +1. So it is now more negative. The outside is now more positive. There's a difference in charge. This is an electric current. So how do our bodies create electric currents? They move pluses, 3 pluses out, 2 pluses in. So 3 sodiums out, 2 potassiums in. And when this happens, you have a difference in electrical charge, and that is how your body becomes electrical. This is why when someone's heart stops, they shock the heart so that you can get those muscles to trigger and start beating again. You need the electric current shocking the muscles in order to make them work. And your heart is a muscle. This whole process, for this to work, it requires energy. And why does it require energy? You're moving sodium and potassium against their concentration gradients. So therefore, that requires energy. And this protein actually has to conform and change shape to do it. So endocytosis. All right, let me slide this over. Endocytosis. All right. Now, cytosis and endo, they go together. This means taking something internally, endo, and cytosis is engulfing it. So something is getting engulfed and brought to the inside, to the endo. So this is the process in which cells engulf bulk or large items. They engulf things, basically. So what's happening here? Here's what's happening in this picture. There's two types. There's either phagocytosis or pinocytosis. So let's talk about the differences, all right? So what happens? In phagocytosis, you're talking about larger particles or whole cells, all right? So larger particles are getting engulfed, all right? So what happens? Well, that large particle or that large cell, this is phagocytosis, comes across the cell that can do this. This could be like an immune system cell. The immune system cells then, and basically their membrane surrounds the item. So they create like a pocket in their membrane, and it surrounds the item completely, and then it pinches off into what's called like a vesicle. Remember vesicles from our last chapter? These are membrane bags, basically. What was that membrane bag made out of? It was made out of the plasma membrane that surrounded the larger particle, in this case, causing phagocytosis to take place. So this is a large particle, gets surrounded by the membrane. This process takes energy. The membrane then pinches off, and now that membrane is a vesicle containing the object that was swallowed up. This is phagocytosis. Pinocytosis is when a cell, for example, is engulfing up smaller items like solutes, things that are dissolved in a solution, or fluids, for example. Same type of thing. They come across, the membrane pinches around them, and then eventually it pinches off, and then it's released, and now it is a vesicle. So pinocytosis is endocytosis of smaller items. Phagocytosis is engulfing larger items. This is considered active transport because it requires energy. So this whole process takes energy to wrap a membrane around something, basically. All right? And this is just showing you more pictures of how this happens. The particles enter, it wraps around them, it pinches off, and now a vesicle forms. Here's another picture showing this is an immune system cell engulfing a bacteria, which is pretty much what's probably happening here. It engulfs it, it enters, and then boom, it breaks it down. Inside of it, it has lysosomes that can now break down what's inside of the vesicle. The reverse of endocytosis is exocytosis. This is releasing items. So, for example, we talked in our last chapter how the Golgi modifies, packages, and ships things. And it ships them in vesicles. So how does it release them from the cell? Well, the vesicles move towards the membrane, the plasma membrane. They fuse with the membrane because the vesicle is made out of phospholipids, just like the membrane. So the phospholipids and the phospholipids, they come together, and then it becomes part of the membrane, and then it opens up, releasing the particles from the cell. Exocytosis also requires energy to do this, to move these items. It is a form of active transport because it requires energy, and it is very simple. It's just the reverse of the endocytosis. All right. So we have three types of active transport. Let's go back and review them. Active transport going from low to a high requires energy. All right. This is showing you one type of active transport, a pump. Here's another specific, cell membrane pumps. In this case, we have the sodium-potassium pump. It moves 3 sodiums out, 2 potassiums in. This creates a difference in charge, and the difference in charge is how our body creates electrical charges and how our nervous system works. And finally, endocytosis engulfing smaller items, pinocytosis, phagocytosis, larger items. Once again, they get put into vesicles or bags when they enter the cell. And again, exocytosis, the vesicle goes to the membrane and gets released out of the membrane. All right? That'll conclude today's lecture.